Hello everyone, my name is Jessica Cuartero Moñino. I am a PhD from the University of Cartagena, and I am going to talk about the results of my PhD thesis through this presentation called Exploring Microbial Communities through Bioinformatics. First, we must know what an environment system is. The environment can be defined as the sum total of all the external forces, influences, and conditions which affect the life, nature, behavior, and the growth, development, and maturation of living organisms. For that, we must study all the factors, but also their interaction in order or with the goal to understand a complete environment. In my case, I am going to focus in soil environment. The soil environment can be defined as the variety of physical, biological, and chemical factors that affect the abundance and diversity of microbes found in the soil. And perhaps you're wondering why we should study the microbial community, and we must study because they perform a key role in soils. For example, soil microorganisms takes part in soil health, but also in soil quality. Both of them are diminishing in their past years, maybe by different factors like human activity, but also climate change. And it is expected this trend continue growing in the coming years. Soil microorganisms perform different tasks in soils. For example, they contribute to soil structure, organic matter renovation, toxin accumulation, but also elimination, and it also contributes to soil-borne pathogen suppression, so it could protect the crop. However, the study of microbial communities is a challenge nowadays because per 1 gram of soil you can find billions of microorganisms, so it's a crazy, with thousand species. Most of them are uncultivable. However, the development of NGS technologies has changed this, and now per 1 DNA molecule you can get all the taxonomic classification of these microorganisms. Currently, I think the most common techniques used are the shotgun analysis for study the metagenome, but also the amplicon sequencing for DNA fragment amplification. For example, 16x for bacteria, but also 18x for fungal community. This is an example. In general, the shotgun analysis offer a higher accuracy than amplicons because you can get fragments with 1,000 base per fragment against the 300 to 500 fragments from amplicon sequences. In addition, you can also get the functionality from shotgun because you have all the genome. Of course, you have, for example, the protein codification, and you can get more information if you have all the genome. But you can also get predicted functionality from amplicon based on their classification. However, the shotgun analysis is expensive and it requires a high computational research. However, the amplicon analysis is faster, is cheaper, and maybe you can perform your own analysis in your own computer. I am going to focus in this kind of technologies because I use that in my thesis. So when you perform an amplicon sequences, per one sample, you can get thousands, millions, and maybe billions of sequences, and you must process these sequences through bioinformatics. Of course, I know you probably know how to process this kind of sequences, but I am going to give you a little steps. The first, we must know the FASTQ file, the different parts they have. We found, for example, the header. It is a unique codification for each sequence in your sample. The second one is the sequences of our microorganisms. And the last one is the quality score. So you can perform, for example, a quality filtering based in these sequences. Of course, I am going to talk about the Kind2 platform. I know there are another different platform, for example, Motor, or maybe you can use, for example, the mgrad server, but I use this. And the first step is import the sequences to the platform, and then you can perform the denoise. Of course, how about the clustering step? 2 years ago, the clustering was a mandatory step because you have to remove the PCR errors. However, the development of different algorithms like the DI2 has changed this, and now you can find publication with both OTU but also SVA. So now, in my opinion, this is a personal choice. But in the future, probably, at least in soil environment, you will find more publications with HPE because they have, or you can get a higher diversity compared to ODU. Regardless the kind of sequence you chose for your analysis, the next step is perform the taxonomic classification. In my case, I use for bacteria community, I use the CEBAS database, of course. There are another alternative, for example, green genes. And for fungal community, I use UNIT database. When you perform the taxonomic classification, you will get something like that, the different taxonomic rank, for example, phylum, class, order. Maybe it's not very good for a species level, but you can also get one of them. And an additional step, for example, to get the functional prediction. Of course, this is a prediction based on the taxonomic classification. For that, you could use, for example, the PICRUSt algorithm, or maybe for fungal community, you can use fungal traits. Bioinformatics is an interdisciplinary field of research to analyze large datasets from TS technologies, which is based on disciplinary knowledge of not only biology and computer science, but also statistics. So statistics is very, very important for bioinformatics because you can explore your data, you can analyze your data, and of course, you can comprehend your results. Well, during my PhD, I read a lot of different statistical methods and techniques, and I performed my own personal classification. For example, statistical methods for analyzing experimental data. Soil properties is an example of that experimental data. Statistical methods for analyzing sequencing data, for example, for amplicon. And statistical methods for analysis both experimental but also sequencing data. Regarding the first group, you can only analyze one variable or individual properties. So for that, the first step is to study or test the normality of the data. If you don't meet the normality of the data, then you should perform a non-parametrical test like Kruskal-Wallis test. If you meet the normality, then you must test the homoscedasticity of the data, and if you don't meet the homoscedasticity, then you have to perform an alternative test like one-way test. But if you meet both of them, normality but also homoscedasticity, then you can perform a parametrical test like an ANOVA. However, if you don't meet the normality, but you meet the homoscedasticity, that you can, of course, you have a very high number of samples, you can add a bootstrapping algorithm and you can correct that normality. Well, if you want, for example, study several properties at the same way, you can use multivariant techniques. So I found that the Pillay-Asankar classification is very good for this kind of statistical methods, because they have a group for exploratory data or statistical methods, for interpretative methods, but also discriminatory methods. In the first group, you can find, for example, the PCA, probably you know. In the second one, you can find, for example, the PermaNova, but also ANOSI. And in the last group, you can find, for example, the random forest. The random forest is a machine learning algorithm, and you can perform a classification, but also a prediction Well, regarding my own second group is the sequencing data. The first is to study, for example, the alpha diversity of the microbial communities. But another option is, for example, study the relative abundance. You can perform this study, this analysis at each level you have. For example, in this case, you can use phylum, order, class, whatever you want. You can also study the beta diversity to see the change between, for example, if you have two cropping systems, two treatments, you can see the difference in the all microbial community. However, has been developed different algorithms, and in this case, I want to show you the left algorithm because it is a special algorithm for this kind of data to get a potential bioindicators. This program is available for free in the Galaxy platform, and you can use for your own project without any cost. And this algorithm performs the first step is the Kruskal-Wallis in the class. The second one is the Wilcox-Ransom in the subclass. And the last one is a linear discriminant analysis. Finally, you will get, if you have in your samples, you will get a potential bioindicator, and you can show the results through these kinds of charts called cladogram. In addition, of course, probably you want or you will want to study the relationship with those microorganisms in your samples, and maybe you can use the MINA algorithm. This algorithm can construct a full network based in your sequencing data. In the network, you can find, for example, the nodes. The nodes are each microorganism in your sample. The S is the relationship between two microorganisms, and the module. The module is a group of microorganisms. When you have your network, that you can, for example, study the topological roles, the characteristic of the network. You can also detect, for example, different groups of microorganisms. Maybe you can study for example, the effect of your soil environment, your environmental in your community, or maybe you can deepen into the modulation. And of course, you can compare. If you have different cropping systems, you have different treatment, you can compare and see the change between the network with the microorganisms. In addition, you can also classify the microorganisms in 4 different roles based on OLSM classification. You can find, for example, peripheral microorganisms. We have a very low number of connections. The connectors are microorganisms which have a higher number of connections, but inside its own module, inside its own group. The network hubs, they are microorganisms with a lot of connection, many connections inside its own group, but also outside its own group, but also inside its own group. And the last one is the module hub. The module hub, they have a higher number of connections, but outside its own module. Well, now I want to speak briefly about for example, the techniques you can use if you want to analyze together the microbial properties and also, in my case, soil properties. And you can use, for example, the correlation, Pearson or Spearman, different functions like bioinf or mfit, but you can also use the mantle test. Well, I have finished this kind of presentation introduction, so I am going to talk about the results of my thesis. I have published 4 different publications, but I am going to focus in 2 of them because I think maybe it's interesting for you, because I perform it's the same experimental design, but I changed the bioinformatic pipeline. Well, regardless the experimental design, We studied the effect of 3 different cropping systems. I remember I am agronomic. So we studied first the conventional cropping system with fertilizers and pesticides. The second one is a sustainable cropping system with compost and compost tea. And the last one is a cropping system also sustainable with manure. It was a long-term experiment. It means more than 10 years old. The plots were located in Cartagena, is a city in the south of Spain, and we performed a crop rotation. It means different crops each year. We had a total of 5 samples per cropping system, and the sampling time was in February 2018. Well, regardless the publication, in the first one, we explored in general the microbial communities, and in the second one, we explored the relationship with those microorganisms, but also their potential functionality. This is the results of the first publication. I am going to talk about the alpha diversity, a common step in this kind of analysis. And we found, for example, how compost increases the bacterial index diversity, but in fungal communities was the manner which increases the fungal diversity. We also studied the beta diversity, and we found differences with the cropping system on bacterial community, but the difference was very higher in fungal community, as you can see in the chart. We also studied the relative abundance. In this case, it's at phylum level, so it's usual, not very strong difference in this case. And for example, we found a little difference in planktonicity in the case of bacteria, but also in, for example, bacterotetes. However, in fungal community, we found several things, especially in cropping system with compost. For example, we have a higher content or Orpiromycota, but also we don't find Glomerocomycota. Well, we use left to find a potential bioindicator for our crops, and we found how, for example, in the conventional cropping system, we had a higher content of potential bioindicators, all of them related with pesticides and herbicides, zoological. In the cropping system with compost, for example, we found a Turicibacter. This is a genus related in soil relationship with soils with high content of organic matter. And in the last cropping system, we found Alliangium. Alliangium is a bacteria which can produce antifungal compost. So in agronomic, it is very interesting because maybe he can control the other pest. We also we try to discover the influence of the soil properties in our bacteria community, and we found how the compost and the soil properties in the compost cropping system highly affect the bacteria community compared with the other cropping systems. Well, now I am going to talk about the results of the second publication. The first, we get a network. If you perform your network, you will get the result of the empirical network. It means the data, your own data, the result of your network. But the MINA produced 100 random networks. So you can compare with your cropping system using random networks. I have selected different topological parameters. For example, the average path distance is the distance between microorganisms. The average clustering coefficient, this is the capacity to perform a cluster. But also the modularity. The modularity, if you remember, is the groups of different microorganisms. Well, this is the network I get from the conventional cropping system, and I found a total of 13 modules, and I found only 2 correlations between those modules. However, in the cropping system with manure, you can see we get only 12 modules, and we didn't find any correlation between them. In the last cropping system, when we added compost to the soil, we find, however, 15 modules, and we found several correlations with them. So this kind of network or these parameters indicate a higher modularity and modular behavior. In addition, each class of network can fully adapt to environmental change. So the network has or presents a higher habitat heterogeneity, no random interaction patterns, and also ecological network complexity. Well, to deepen into the relationship between those microorganisms, we selected the 10 most abundant microorganisms in our samples, and we performed a network. In the conventional cropping system, we found, for example, a higher number of negative connections in the subnetwork, but we found a higher number of positive connections in the global network. In the cropping system with compost, we found a higher number of positive connections in the subnetwork, and in the global network, we find a higher number of negative connections. The positive connections implies cooperation and codependence between nodes, between microorganisms. However, the negative connections implies rivalry, and which could lead to an stabilization of the network. We also studied the different roles of the microorganisms, and we found how the cropping system with compost had a higher number of generalists, it means connectors. The generalists are the key microorganisms and play important roles, especially in soils, because they have a higher environmental tolerance. So it could be interesting, for example, for climate change. Well, now for finish, I am going to talk about the potential prediction, but I selected only the nitrogen pathway. And I am going to talk, for example, the nitrogen fixation pathway, and we found how the compost increased the NIF gene. In the nitrification pathway, we found also the gene RNFE, and the no-zeta gene was also increased when the compost was added to the soil. And in the last one, we found, for example, how in the nitrification pathway we had a higher content of how gene in the cropping system with compost. So this is my presentation. I hope you understand everything. Thank you so much.